Thank you for your invitation. I'm just going to go for your kind introduction. So I'm a clinician. So the way I give this talk is to make it more like a journey that I went through, which would be more, I felt like this sort of talks would be more interesting for students, more younger generation who want to go through this research path and, share with them the obstacles and, hopes and excitement, etc. So, typically in a clinical setting, what we do is that we cut down the immunosuppression, try to reduce the toxicity. But what happens most of the time is that the rejection takes place. So most of these trials that we've done to replace one drug versus the other one hasn't been that successful. So I got across with the term nanoparticle many years ago, and I was talking to my engineering colleagues. They were talking about drug delivery, and I said, that's a pretty interesting concept. Can we deliver immunosuppressive drugs to specific sites? And at that time, most of the trials, most of the efforts were gone into the tumor, because 95% of the efforts in nano-targeted delivery was to get the drugs to site of tumors. And, you get a number of benefits from packaging a drug into a nanoparticle. But I think most people are interested in this one, where you can get the specific drug to a specific site in body. This slide pretty much shows some of the issues with nano delivery. Basically, this is how it started. It is a concept called passive targeted delivery, where the vasculature of tumor is thought to be leaky. So you make the particle drugs inside the nanoparticle and inject intravenously, and the particle home passively to environment of tumor, because the vasculature of tumor is disorganized and leaking. But we learned more and more that actually this is not the case. A lot of tumors, the tumors that are so-called, especially cold tumors that don't respond to therapies, they're actually quite fibrotic, meaning that the leakiness is not the case. So more and more people go after so-called target delivery, where basically you conjugate the surface of nanoparticle with antibodies or some kind of peptides that recognize certain receptors on your targeted cells. It could be, a cell in the kidney, in the liver, but the rule of thumb is that the receptors that you're interested to target should not be widely expressed, because then you're going to target a large number of cells, different tissues. So, but most of the time, I would say, people use antibodies on the surface of this particle, put a drug inside, but you can, put different sort of targeting that can be applied. So as I show you, one of the critical things to remember is that the environment is not as leaky as we think. See, this is the vasculature that, let's say, you're hoping that nanoparticle is going to pass through. So in the case of, for instance, tumor, or in the case of transplant organ, there's a significant amount of ECM is built around the vein, around the vasculature. Here is the fibronectin, is basically shows fibrosis. And if you cut your skin and you have a big scar, you're going to see massive accumulation of fibronectin. So this is a tumor microenvironment. You see how much fibrotic material has been made. So the passive delivery is not as easy as we thought. It's not like a prevailing theory anymore, because even in the case of tumors, there's quite a bit of materials made. And there's been studies actually in the past that if you inject an antibody, less than 1% actually gets to the tumor. So let's say this is a tumor environment and you inject an antibody with a drug. Antibody needs to pass through here and pass through here to get this tumor. When it comes about drug conjugation, it's a very easy concept. You say, I made an antibody that recognizes this tumor and sticks to these tumor cells, for instance, here, and then brings the drug inside. But most people don't talk how this antibody gets from here to here, you see. So this is a very, very important angle to think about it. That's one of the problems with the drug delivery, is that most people don't pay attention on this path, that this big molecule, the antibody, needs to pass through this vasculature. So we became interested in two sites in transplant. So in transplant, so this is an organs that is being transplanted in a patient. So it comes with those own antigen presenting cells. These are the cells that present antigen. They reside in the tissue, donor tissue. And when this antigen basically of donor antigen is being recognized in the lymphoid tissue of host. So remember, immune education takes place in lymphoid tissue, doesn't take place in blood, right? So that's a very critical point to get across for engineering people, that most important site of immune activation is in the lymphoid tissue. So these are two sites that most people don't study actually, because most people are like to study tumor itself. That's another obstacle. I realize that, because 90-something% of people are interested in getting the drug to the tumor, right? And the reason for that is because most people who started in nanodelivery came out of the cancer school, and they've been taught that if you get the drug to cancer, you get better outcome, which makes sense. But we know more and more that the lymphoid tissue is a primary site of tumor immunity, right? So if you're able to get there, you can change the immune response against the tumors or transplant. So one of the interesting things about lymph nodes that they have a very special vein, which is called high endothelial venule here, shown here, where which express this molecule, which is very interesting and exclusive. It's called PNAD, peripheral node addressing molecule. And this is a sugar-coated molecule that expressed on this class of vein. And me and Pilhan's group has been working quite a bit on this vein for a number of years now. And so this vein, you're born with it in your lymph node. So you in your 200-300 lymph nodes, you have this vein. And the reason they're called high endothelial venule, or I'm going to call from now on HEV, is because they have so much Golgi and the plasmic reticulum to make this very complicated sugar-coated glycoprotein. Basically, we want to make a nanoparticle here and put the drug inside the nanoparticle. And on the surface, we're going to put this antibody, which is called MECA79, which I told you that recognizes this PNAD. So the nanoparticle binds to this PNAD, gets internalized, and releases a drug in this neighborhood. So that's the concept. And the drug of choice here is the basically mouse. We put the skin transplant on the back of the mouse. And typically this lymph node in a mouse gets really big. You don't need to have a microscope. The size gets huge post-transplant. And we show that we put an infrared dye in this nanoparticle, and this particle had the antibody on the surface. And you can see these lymph nodes become hot. This is a draining lymph node. And if you examine the sections of this lymph node over time, this is the HEV structure of a lymph node. Few hours are mostly in the HEV complex. And as time goes by, this particle migrates out in the lymph node into the HEV. So when we did more staining, we find that this dye sticks around for 3-5 days. So it's very hard for us to understand the kinetics of the drug, meaning that you have to do HPLC or very fancy stuff. We haven't done those things, but it seems that the average half-life of this particle is about 4 or 5 days, because these are biodegradable particles that breaks down. So this round, we said, well, we want to put a drug that is more clinically applicable. You see, in that drug that I told you about, tacrolimus, is an immunosuppressive drug that you take for the rest of your life, right? A transplant patient takes it every day, twice a day. So that's a cool science, right? Because you say that, but you know that clinically is not going to happen, right? I cannot give nanoparticle to a patient every day unless this causes magic thing, right? So when you, again, if you want to increase translation, you need to think about how this product going to be used by physicians, because if you say that you need to take this nanoparticle for a long period of time, they're going to say, well, my patients even don't take their own simple pills. I'm going to take them to come to clinic and get this nanoparticle. So we said, well, we want to do a practice or do a drug that used for 4 days, 5 days. So this class of drug called depleting agents, this is called anti-CD3, which is an antibody that depletes immune cells, basically wipes out all the CD3 cells. And they're called depleting agents. And they're actually very commonly used in a wide variety of immune conditions that are lethal, like let's say lupus patients or diseases that, or bone marrow transplant, diseases that you really want to give a lot of immunosuppressive drugs for a short period of time. And we call it induction, meaning that you induce significant number of immunosuppression and then put them on maintenance, like tacrolimus. So if you come say to a physician, instead of giving like, I don't know, 2 milligram per kg of this drug, and a large number of patients get infection, malignancy because they're very toxic, or, I can cut them by 100%, but you have to give this nanoparticle 3 times only. It's clinically feasible. So the reason we went for this drug was that we want to make sure something that is clinically more feasible. So the concept was the same. We put our antibody inside the nanoparticle, and outside of it, we put our antibody, which is a MECA79 here. So in the case of free drug is this one. Remember, as I said, this mice rejected on day 10. So if you give the free drug, it prolongs it very nicely. You see, it brings it from 10 to almost 30 days. And if you give this drug with this particle, it significantly prolongs. So, and the injection was about only few injections here. Wasn't giving for all time. And this drug, remember, it causes tolerance, meaning that it doesn't need to be given every day. And, more recently, we actually went through other classes drugs, which are called co-stimulatory blockades. And this is work that we did with Pilan. And the same concept, these are newer class of immunosuppressive drug, but that if you're able to get them to the lymph node, you get better tolerance with lower drug needed. These are various forms of human tumor that you can see this way. This is a pancreatic tumor, which particularly shows quite a bit of expression of this vein. For some reason, we're trying to figure out. We have a paper with Pilhan specifically on mechanism of induction of this vein. And you can see across all tumors, they have it. And the reason people more and more becoming aware of it, because they put antibody to it, right? Because the people are getting interested in this vein. Remember, this vein does T-cell homing. So, and checkpoint inhibitors and, tumor immunity is very hot now, right? So if you know how to make this vein, the idea is that you can get more T-cells, right? And then that be a major cornerstone for T-cell, for tumor immunity, right? Because cold tumors don't do T-cell homing and they're cold. So one of the things that we found, which I think is quite amazing and it's not being still appreciated, is that metastatic site also makes it, right? So this is known. This is quite widely published in the case of primary tumor. But this is the metastatic site of a patient who died with breast cancer. The lung actually has more of this high endothelial venule than the primary tumor. I don't know why. Maybe because when they go to the distant metastatic sites, maybe they have more progenitors there. And maybe those sites have, cells have be more prone to metastasize and maybe have more enriched with HEV. We haven't quantified it, but it looks like that way. Here is a patient so we see basically the concept that we are pushing or we are setting forth is that you have primary tumor that you have HEV, you're born with lymph node that you have HEV in it, and you have metastatic sites that seem to have HEV. So if you target HEV, you may get triple site delivery. We basically put the tumor in an NSG mouse, the pancreatic tumor that had the HEV, and we made our own particle, and you can see that mouse that had the pancreatic tumor that received MECA79 infrared dye is hot now. So we showed that this tumor, actually, you can get the nanoparticle to it. And that time we put a Taxol to it, and group that received chemotherapy with the MECA79 MP, they had much smaller size tumor. So we showed that the particles home to the tumor, and you can get smaller size. So this is the concept that we are promoting, is that you have particle that sticks to this primary HEV, which has the PNAT, right? Because you have it, you're born with it. And with this, you can actually hit the tumor drain lymph node. Then you have primary tumor that express the same molecule, and you have distant HEV that has the same molecules. This basically explains the so-called triple site delivery that express the same molecule. And although the structure of HEV might be different, that's why people were interested in these two things. But you should remember that the HEV doesn't look like classical HEV. So if they stain it and start seeing the structure is not as beautiful and bubbly looking, chubby, fat ones, remember that this size, the HEVs are not well organized. So, but they do express the same molecule here in the case, non-nanoparticle, and this is a breast tumor, which actually expresses PNAD, because this is a newly made and high endothelial gain in the tumor. And the mice that received the Taxol MHA-112 versus free drug had much smaller size tumor. For the kidney, we made a model where we targeted the receptor on the proximal tubular cells called megaline. And in this case, actually, we put a light chain, because in your body, light chains are produced more and they excreted by kidney, but they're pecked by a proximal tubule expressing this molecule called megaline, and goes inside the proximal tubule and breaks down. So in this case, we actually didn't use antibody, we used light chain. And this is a route to get to the kidney. It could be an interesting route for this is the proximal tubule for kidney cancer. Could be an interesting route for treating immune conditions of kidney, like lupus nephritis, for instance. This is actually an interesting, because in no other places you do that, right? You don't go in a patient, take the liver for liver fibrosis, right? Treat the liver and put it back, right? So transplant is the only condition you have. So this is a fascinating situation where you can actually do a lot of things to the organs prior to putting to the patient. So one of the big obstacles that we have is that when you remove an organ from a donor, that time donor organ is inflamed. So one thing we could do, we can actually treat this with nanoparticle inside these organs and suppress the inflammatory responses in these organs by putting nanoparticles to these organs. And this can happen prior to transplant. So this route is the most effective way to protect the organ in the host. But what we do now is that we put the organ in a patient, right? And we give this patient a lot of immunosuppressive drugs that are toxic. First, again, this is another problem. We don't know how much drugs get to this organ, where basically is the site of inflammation now, because all the T cells and macrophages want to go there, right? So if you have the organ already with nanoparticle, that drug is being released, you get a protective effect from day zero against inflammation. So this is a very interesting, important concept. One of the drugs that we are very much interested in is the anti-IL-6, because we are thinking that IL-6 is a significantly pleiotropic cytokines that causes massive augment the immunity against the transplant rejection. And they do it in different ways. It's not just one way. So if you target this pathway in the organ prior to transplant, our hypothesis is that you get much better protection. Here, for instance, we give local, meaning that we just give the anti-IL-6 to the organ and compare it with when we give it with systemic anti-IL-6, meaning that either give significant above drug to a host IV or give much smaller amount to the heart transplant prior to transplant. So you either give it to small amount to the organ, protect the organ, or give huge amount to the host. And we found that actually through this route, meaning through nanoparticle route, we get much better protection with much less anti-IL-6, which better efficacy and less toxicity. So here we're collaboration. We have the MGH, and we're trying to do some of these large animals. This is the lung transplant. This is a very interesting tissue because it has quite a bit of macrophage in it. So we are pre-treating these lung transplants with nanoparticles and trying to study the trafficking and kinetics of the uptake of these particles by macrophage. So the pre-organ transplant nanoparticle delivery, the target doesn't have to be transplant rejection. One of the problems that we have with transplantation in general is that we actually discard quite a bit of tissues. A large number of organs being discarded because their quality is not good. They are ischemic, or they've been in ice cold for too long. They have thrombosis, et cetera, et cetera. So, our liver, for instance, there are no massive number of livers are discarded because they're non-metabolically good. So one of the cutting edge idea is that you treat these organs. You basically make a center which you can modify these tissues and then transplant them. You follow what I'm saying? So this is a very interesting concept to your questions. I think I want to also thank again Pilan with this team, and it's been a great collaboration. And I think I told Pilan that maybe one day I show up in Korea, but increase our collaboration. And this is a place that if students or people who are interested in interacting, can always join us, and we welcome, people from Korea and increasing our collaboration. Thank you.